Okay, Jovan. So here it goes for kind of a description of how we're going to do this mutagenesis. So yeah, in lecture today, we're going to be talking about one mutagenesis method that allows us to make a mutate one or multiple mutations in one single site on a plasmid. So we could do that, but then basically we'd have to make one of the one of the mutations confirm that we've made that mutation, and then go through a whole second round of making mutations to make the second one, and again confirm that. And each one of these rounds can take multiple weeks. So instead, I've been looking around to try to see if I can find a way to make multiple mutations simultaneously, and I found a way that allows us to do this. So, all right, the first thing I want to do is just real quickly explain PCR to you. All right, so if we have this big old piece of DNA here and it's double-stranded, right? All right, the way that we do PCR is we basically design primers that are going to anneal to both strands, or to the opposite strands of DNA, and are complementary to, this end of the gene and this end of the gene. And so these end up basically delineating the piece of DNA that we're going to be amplifying. And so our primers, they always go in the 5' to 3' direction. So, once they anneal to our template, DNA polymerase can basically extend on down this way, and then likewise the 5' and 3' ends of this primer, DNA polymerase can extend that strand down that way. All right, so kind of move in these two different directions. All right, and then after one round of this, all right, what we end up with, we have our same pieces of genomic DNA that we started off with, but we've now got these shorter fragments that extended off that way, and another fragment that extended off this way. Right now, with these fragments, again, we can get primers annealing here to this one, but on this one, for instance, all right, we'll have a primer anneal here and extend that way, right, and these end up, this part right here will end up making our gene now, amplifying that one portion of DNA. Likewise, a primer can anneal to this strand here, which will extend that way, or to this strand here and continue extending past the gene. But this product right here, again, then will be our actual gene. So this is a quick, dirty explanation of how PCR works. Now, why am I telling you about this? So we are actually going to use PCR in kind of a creative manner to introduce multiple mutations. And so what we are going to do, we're going to take advantage of a couple things. All right, one is that when we're designing these primers, so one part of the primer has to be complementary to our gene in order for it to anneal to it. But on the 5' end of these primers, all right, we can basically attach whatever sequence we want there, right? Because we aren't going to rely on that portion of the primer to anneal to our to our gene, and the extension can still occur in the 5' to 3' direction, even if this portion up here isn't annealing. Now, eventually, what we will end up with, and if you want to talk about this more, you come and see me at some point, is if we add on these extensions, we end up with our gene of interest with those extensions that we added on being turned into double-stranded DNA on either end of the gene. All right, now, how does this help us? Well, in your gene for SET, we have 2 different mutations we wanted to put in. I can't remember the exact numbers, but one of them is in the middle and one's at the very end, right? All right, so we have these 2 different sites that we want to mutate. Well, what we're going to do, what I'm going to have you do, is basically break this gene up into 2 portions. All right, one portion that's going to be there, actually kind of 3 portions, we'll say. All right, so we'll have portions 1 and 2 here. All right, this part here I'm going to call our overlap region. If I could write. All right, so this is our overlap region. and then we'll treat this end here with the mutation, we'll treat this as an overhang. All right, so what we are going to do here is we are going to separately amplify regions 1 and 2. So what will this look like? So what we're going to do is we're going to design primers that anneal exclusively to region 1 here. So we'll have one primer that anneals up here, and then the reverse primer will anneal over here. Now, I'll talk about the addition. There's going to be an addition on primer number 1 here. I'll talk about what that addition is in a little bit because it's kind of a little side thing for that we're going to use later on. But on primer, the reverse primer, we're going to have a overhang here, and we want this overhang to be complementary to the overlap region, and it's going to contain our mutation. So our mutation, we're going to insert this, our one mutation, into this overlap region, this overhang on the primer. All right, now, similarly, we're separately going to amplify region 2. All right, so we're going to have one primer that anneals up here, one primer that anneals on the back side here. All right, the forward primer for region 2 is again going to have the overlap region with the mutation. All right, and the tail that's hanging off here at the end is going to contain our second mutation, since that mutation's right at the end of our gene. All right, so our second mutation is going to be on that overhang. All right, so the product that we're going to get now from this, all right, so we're going to have region 1 with this overhang, and there's going to be this overhang here that I haven't talked about yet. And so we have our mutation in here. And then we're going to have region 2 with, it has the overhang here again, having the mutation in here. And our second mutation is in this region. All right, now, how does this help us out? So we've made the mutations, but we've basically broke our gene into 2 different fragments here. All right, so I'm going to go to talk about this now. We're going to talk about an advanced form of PCR called fusion PCR. All right, now in fusion PCR, what happens is that we're going to use overlapping regions of our template DNA as basically primers. So here's our region 1 with the overhang. I'll kind of draw it here. All right, gene 2. Looks like so, and it has overlap region there. Now, if we go and do PCR with this, all right, so what we're going to do within the PCR reaction is we're going to combine fragments 1 and 2 both as templates, and then we're going to have primers that anneal only to the forward side of fragment 1 and the back side of gene fragment 2. Now, what are the different things that can happen here? Well, to start with, I'm just going to focus on what's going to happen with the PCR products, because once we understand that, then the addition of these 2 primers will make sense. So, in PCR, we denature the 2 strands of DNA, and then we start to anneal them. So, from gene 1, we can separate the strands like so. We have this as the 5' and 3' direction, and the 3' end of fragment 1 is complementary to the 3' end of fragment 2. So the two templates can actually fuse with one another, and DNA polymerase can extend and extend. Well, what's the end product of this? Well, now we've actually fused together our fragment 1 overlap and our fragment 2. And then it has, the second mutation at the end. All right, so now once we've done this, so we now have these two fragments combined together. All right, the addition of these two primers at the very ends makes sense because we can now use them to amplify and make more copies of this fused gene. All right, so now as far as the specifics of what you're going to have to do here to design your primer. So I'm going to be sending you a document along with this that goes through and talks about how to do primer design. Now, the first part of primer design, so some of what's said in that document is going to apply to this situation, some is not. All right, so the basic part of trying to find a sequence of DNA that will anneal to your gene, all right, that part is going to be the same. All right, except instead of having it, so at the very first part of our gene, all right, we're going to choose somewhere between 16 and 25 bases here from the first sequence of the gene. All right, and in that region, so basically you want to try to select a length and use a melting temperature calculator that I talk about in the document to get a melting temperature that's somewhere in the 65 to 72 degrees Celsius range. All right, so if the melting temperature is too high, well, what we'll do is basically remove some bases off of the 3' end here. Right, we need the 5' end to always be the same because that delineates where the amplification occurs. All right, now you're also going to do the same procedure at the end of region 1 here. All right, so you'll select here 16 to 25 bases. If we need to remove ones or add them on, you do it from now the 5' end of the sequence. So I'm thinking of, real quickly here, I'm talking about having an anneal to the same strand initially, because I think this is usually easier to think about and deal with. So I usually will think about it, designing the primer initially for this strand, for the same strand as this. So that way I can use one sequence, one gene sequence to deal with here. Now, once I've done this, all right, the forward primer, I'm going to kind of discard it for a second. Write down the sequence, but cast it aside for a little bit here. For this region right here, what we are going to do is we're now going to add on the overlap region. So in this overlap region, it will contain our mutation, but it also has to be substantially long so that there's a significant overlap between regions 1 and 2. So in this region here, we're probably going to have, I think I'm going to design it so we have 25 bases in that overlap, and there'll be a third document coming along that has the entire gene sequence and the overlap region kind of spelled out for you here. All right, now, once we have made this primer, we actually need to anneal to the opposite strand of DNA. So what we do is we take this and make something called the reverse complement of it. All right, again, the primer design document I sent has a link or information about how to make reverse complements by using available online calculators. All right, now we're going to make these two primers here. All right, now we also have to make a separate set of primers for region 2. So we'll have one region here that anneals to the front end of region 2, and another region that anneals to the back side of region 2. All right, now the front side of region 2, this front primer, we're going to add on the exact same overlap sequence that we used for the reverse primer for region 1. All right, so this will, both fragment 1 and fragment 2 will have containing that 25 base overlap with the mutation. All right, now on the back side of region 2 on this primer, all right, we're going to add on an overhang here that includes the end of the gene and our mutation, and one little extra sequence at the very end that I'm just about ready to talk about now because it applies to our very forward primer and this very, very end one as well. All right, so, and then again, this primer, once we're entirely done with it, we'll make the reverse complement of it because we need it to anneal to the opposite strand. Now, these overhangs on the very forward primer and the very, very end primer, they also have to contain a restriction site for our gene. So the restriction enzymes are basically allow us to chew off the ends of our final PCR product, creating sticky ends that we can then use to clone the gene fragment into our gene into the plasmid that we're working with. All right, and the two restriction enzymes that we're going to use based on what have been used in this plasmid already, we're going to use for the forward one here, BAMH1, and the reverse one, we will use XHO1. All right, now the sequences, recognition sequences for BAMH1 and XHO1, you can find these online. Just do a Google search for BAMH1 recognition sequence, and you'll add it on to the 5' end of this primer and the 3' end of this primer before we do the reverse complement. So it would be on the very, very tail end here of the end primer. Now, also, once we add on the restriction sites, we're nearly done. Restriction enzymes don't like to cleave at the very tip ends of DNA molecules. So typically, we'll have the restriction enzyme site here. But on the outside of this, we have to add on about 6 base pairs of random DNA. And so 6 base pairs of random DNA there. And this sequence is entirely up to you. All right, and then likewise on the very end of this one, at the very end of the reverse one, you also have to add that same 6 base pairs of random DNA sequence. All right, now once you've once you've finished with the reverse primer for region 2, now you've added on the mutation site, the restriction enzyme site, and the random DNA sequence. All right, now you can make the reverse complement of that one, and we now have our entire gene, all the primers we need for our gene. So you're designing a total of 4 primers here, 2 that anneal to region 1, 2 that anneal to region 2, and then for the very last amplification, we'll use the forward primer from region 1 and the reverse primer from region 2 to amplify the entire gene. All right. If you have any questions, come by and see me.